Then next one. Degree of polarization depends on charge density of cations. Answer is option A. Then deep sea dives use oxygen with added helium. Why? Now nitrogen and oxygen mixtures were used earlier instead of oxygen and helium. In the earlier it was nitrogen and oxygen. Now what happened when you used nitrogen? So as nitrogen is soluble in the blood, it causes bends or decompression sickness, which are sometimes fatal to the divers. So nitrogen is replaced by helium because it is less soluble in the blood. And when using helium, helium can also mean that divers can take fewer stops on their return to the surface without suffering from decompression sickness. So the answer here is why helium is added to reduce acute oxygen toxicity in deep waters. That is correct. To reduce nitrogen necrosis. Now nitrogen necrosis, also referred to as inert gas narcosis or ruptures of the deep sea and the Martini effect, and it is caused by breathing high partial pressure or concentration of nitrogen while under water. So that is called as nitrogen narcosis. Then to reduce decompression sickness, all of the above. Answer is option D, all of the above. Then next one, hydronium ion is. So it is a hydrated proton dissociation product of water in solution with the structure H3O positive. So answer is option D, that is all of the above. Now next question. Cis-trans isomerism is exhibited by. So it is exhibited by malic acid and fumaric acid. So malic acid is the cis isomer, that is this one, first one. Malic acid, it is the cis isomer of butene dioic acid, whereas fumaric acid is the trans isomer, and both malic acid and fumaric acid, they show geometrical isomerism, that is cis trans isomerism. So the answer is option B. So coming to the next question. Pharmacokinetics and pharmacodynamics are the effects of. Now pharmacokinetics, it is defined as the movement of drug through the body, whereas that is drug on the body, and whereas pharmacodynamics is defined as the body's biological response to the drug. So body on drug. So answer is option A, that is drug on the body and body on the drug respectively. Answer is option A. Then DNA from any cell of any organism should have 1:1 ratio of pyrimidine and purine bases is the basis of. So it is the basis of Chargaff's rule. Answer is option B. Then which of the following is a cofactor for succinate dehydrogenase? Now succinate to fumarate in citric acid cycle is carried out by the enzyme succinate dehydrogenase, and FAD is reduced to FADH2 in this reaction. So answer is option C, that is FAD. Next one, prokaryotic and eukaryotic flagella are driven by. So eukaryotic flagella are ATP driven while prokaryotic flagella are proton driven. So answer is option C, that is proton and ATP respectively. Then bacterial genome is protected from its own endonucleases by. So the restriction enzymes in bacteria function to defend themselves against bacteriophages or invading viruses. Okay. So the DNA sequence the restriction enzyme recognizes is present both in the viral DNA as well as the DNA in the bacteria. So bacteria have to prevent these restriction enzymes from digesting its own DNA, and how it does is by masking the restriction sites with methyl groups. So methylation of DNA is a common way to modify DNA function, and bacterial DNA is highly methylated. So by doing this methylation, it functions to make the restriction sites unrecognizable for the restriction enzyme. So answer is option A, that is methylation at restriction sites. Then most striking example of an unique point mutation is found in the disease. So night blindness, it is caused due to deficiency of vitamin A. Then thalassemia, it is an inherited autosomal recessive disease resulting from mutations in the alpha and beta globin gene clusters on chromosome 16 and chromosome 11 respectively. It is characterized by reduced or absence of synthesis of globin chains of hemoglobin and includes two main types, that is alpha and beta thalassemia. Alpha thalassemia is caused due to deletions or mutations in globin gene. And beta thalassemia, they are heterogeneous, that is they can be caused due to different kind of mutation. That is, they can be either due to single nucleotide substitution, deletions or insertions, or oligonucleotides leading to frame shift. Then Down syndrome, it is due to the trisomy of the 21st chromosome. And sickle cell anemia is due to change in a single nucleotide. It is a point mutation in the beta globin chain of hemoglobin. It replaces the amino acid glutamic acid by valine at the 6th position of the beta chain. So the 6th codon in the beta chain, that is GAA, becomes GTA, and accordingly the 6th amino acid becomes valine instead of glutamic acid. So the answer here is option D, that is sickle cell anemia. Then next one, proteins absorb UV radiation at 280 nanometers maximally due to the presence of tryptophan, tyrosine, phenylalanine, all of the above. Answer is option D. Then which of the following does not have coenzyme activity? Then thymine, if you take option B, thymine, the thymine pyrophosphate or TPP, it functions as a coenzyme for number of enzymes involved in carbohydrate metabolism. Then if you see option C, that is biotin, it is a coenzyme for 5 carboxylase enzymes which are involved in the digestion of carbohydrates, synthesis of fatty acids and gluconeogenesis. And riboflavin, if you see, it is essential for the formation of two major coenzymes, that is FMN and FAD, that is flavin mononucleotides and flavin adenine dinucleotides. These coenzymes are involved in energy metabolism, cell respiration, etc. So the one which does not have coenzyme activity is vitamin E. Answer is option A. Then active transport. Then active transport is the movement of molecules across a cell membrane from a region of lower concentration to a region of higher concentration against a concentration gradient. So active transport, it requires cellular energy. So the answer here is option B, requires energy. The next one, the general name for the enzyme that transfers phosphate groups from ATP to a protein is, so they are called as the protein kinases. Answer is option A. Then Tata box and Prigno box are components of, so they are components of the promoters. Answer is option B. Which of these molecules or complexes have a greater oxidizing potential than oxygen? So it is photosystem 2, that is P680+. Answer is option B. Then next one, the relationship between legumes and rhizobium is, so legumes are able to form a symbiotic relationship with nitrogen fixing bacteria called rhizobia or rhizobia. The result of this symbiosis is the formation of nodules on the plant root, within which the bacteria can convert atmospheric nitrogen into ammonia that may be used by the plant. And the legume plant, what it does is it provides the bacteria with carbohydrate as an energy source. So there is a kind of mutualistic relationship. Both the legume and rhizobium are benefited by this type of relationship. So it is called as mutualistic relationship. Answer is option A. Then next one, a biocycle with period of 24 hours is called. It is called as circadian rhythm. Answer is option B. Both photosynthesis and respiration require chloroplast. Chloroplasts are required only for photosynthesis. Sunlight is required for photosynthesis. Mitochondria is required for respiration because photosynthesis it uses chloroplast. And cytochrome. Cytochrome are required by both. So answer is option D. Then next one. Ribozyme is made up of. So ribozymes are catalytically active RNA molecules. So the answer is option A, that is RNA. Then the stage of meiosis where cells become haploid. So in anaphase 1, the homologs separate to opposite ends of the cell. Sister chromatids, they stay together. And telophase 1, newly forming cells are haploid or n is equal to 2. Now in meiosis, if you see a string of 23 chromosomes separate from another 23 chromosomes. Okay. So and this take place during the anaphase 1, that is homologs separate to opposite ends of the cell and sister chromatids, they stay together. So from 46, 23 going to one pole and 23 going to the other pole. That is what happens in the case of anaphase. So answer is option C, that is anaphase 1. The next one. The excreta of lizards is rich in. Lizard is a reptile and it is rich in uric acid. Excreta will be rich in uric acid. Answer is option B. Then next one. Who demonstrated that genes are located on the chromosome? So it was none other than Morgan. Answer is option A. Then genetically engineered male sterile crop plants have been produced by inserting. So Barney's gene is the answer. Answer is option C. I will tell you. Then Barney's gene expression within the anther selectively destroys the tapetal cell layer that surrounds the pollen sac and prevents pollen formation and lead to male sterility. So Barney's gene, it targets the tapetal cells. Now what is the importance of this tapetum? It is important for the nutrition and development of pollen grains as well as source of precursors for the pollen coat. So genetically engineered male sterile crop plants, they can be produced by inserting Barney's gene. Answer is option C. Then next one. An open reading frame is one that has. So an open reading frame are defined as spans of DNA sequences between the start and the stop codon. So answer is option B, a start and a stop codon. Coming to the next question. Which of the following restriction enzyme produces blunt end DNA fragments? So ECO-R1, 2 and BAMH1, they are all producing sticky ends. ECO-R5 does not produce sticky ends, it produces blunt-ended DNA fragments. Then next one, a high BOD value in an aquatic environment is indicative of. So biological oxygen demand. It is the amount of dissolved oxygen needed by aerobic biological organism to break down organic material present in a given water sample at certain temperature over a specific time period. Now that is the definition for biological oxygen demand. Now what does it say? It is the amount of it is the amount of dissolved oxygen needed by aerobic biological organism. Let us take the example of bacteria. Now bacteria require certain amount of dissolved oxygen for what? To break down organic materials present in the given water sample at specific temperature and over a specific time period. So when the sewage is high in BOD, it means that there is decrease in oxygen. Now decrease in oxygen can be seen if there is excess of nutrients. So answer is option B, that is a highly polluted system due to excess of nutrients. The next one. Greenhouse gases absorb the light of wavelength. Our answer is option D, that is 450 nanometers. Then Western blotting is a technique for hybridization of. So a Western blot, it is a laboratory method used to detect specific protein molecules from among a mixture of proteins. In Western blot, the target proteins are transferred to a hydrophobic membrane after SDS page and then detected using specific antibodies. So here the protein of interest is our antigen. And this particular antigen, it can be detected by a specific antibody which is specific for that particular antigen. So Western blotting is a technique for hybridization of antigen antibody. Answer is option A. Then female gametophyte develops within. So female gametophyte, it develops within the ovule. Answer is option A. Then cyclins are proteins involved in the regulation of. Now cyclin is a family of protein that control the progression of a cell through the cell cycle by activating cyclin dependent kinase enzymes or group of enzymes required for the synthesis of cell cycle. So cyclins are proteins involved in the regulation of cell cycle protein kinases. Answer is option D. Next one. The major site of attack of HIV on immune system is. So it is T lymphocytes or CD4 plus T cells more specifically. So it is the T lymphocytes where HIV attacks. Then photochemical smoke always contains. So photochemical smoke, it is a mixture of pollutants that are formed when nitrogen oxides and volatile organic compounds react to sunlight, creating a brown haze above the cities. And the major undesirable components of photochemical smoke are nitrogen dioxide, ozone, peroxyacetyl nitrate or PAN. So answer is option A, that is ozone. Then development of fruit without fertilization. That is development of fruit without fertilization is parthenocarpy. Answer is option A. Parthenogenesis is the spontaneous development of an embryo from an unfertilized egg cell. It is not the case of fruit. So fruit without fertilization is parthenocarpy and an embryo from an unfertilized egg is parthenogenesis. Then sporogamy is the fertilization of seed plant involving passage of the pollen tube into the ovule by the micropyle. That is sporogamy, and autogamy, it is self-pollination. So the answer here is parthenocarpy. Then next one. Which among the following is a sterilizing agent? So it is ethylene oxide. Answer is option A. Then Shine-Dalgarno sequence in bacterial mRNA is near to. So the Shine-Dalgarno sequence is a ribosomal binding site. It is a ribosomal binding site in bacterial mRNA located around 8 bases upstream of the start codon, that is AUG. Okay, so the answer here is option A is near to, that is Shine-Dalgarno sequence in bacterial mRNA is near to AUG codon. Then next one. Which among the following is not a universal stop codon? UAA, UAG, UGA, all these are stop codon, whereas UAC is not a stop codon. Answer is option C. Then next one. The condition albinism in man is linked to deficiency of the enzyme. So mutations in thyrosinase gene causes albinism, an autosomal recessive disorder characterized by reduced melanin pigment in the hair, skin and eyes. So answer here is option C, that is thyrosinase. Then which of the following anti-tumor agents acts by impairing de novo purine synthesis? So it is methotrexate. this enzyme, agent, it inhibits the enzyme dihydrofolate reductase, which catalyzes the conversion of dihydrofolate into tetrahydrofolate. And this tetrahydrofolate, it is necessary for synthesis of nucleotides of both DNA and RNA. And methotrexate, it also inhibits the de novo purine synthesis of both purine and thymidylate synthase, thereby inhibiting DNA synthesis. So the mechanism is used in the treatment of cancer because of this cytotoxic effect. So answer is option C. Then rRNA originates from the nucleolus. Option. All of the following are proteins within the core nucleosome particle except. So a nucleosome, it is a section of DNA that is wrapped around a core of protein, and this core of protein, they are histone proteins, and this DNA segment, it is wound around 8 histone proteins. So this histone octamer, it consists of 2 copies each of histone proteins H2A, H2B, H3 and H4. So option B, C, D are not the correct answer because they have asked for one which does not form the core of the nucleosome particle, and it is option A, H1 is the answer. H1 is a linker between nucleosomes. 2 nucleosomes are linked to each other by H1. Answer is option A. The next one. Which of the following chromosomal alterations would you expect to have the most drastic consequences? Answer is deletion. Why? Because deletion results in a frame shift mutation that changes the reading of subsequent codons and therefore alters the entire amino acid sequence that follows the mutation. So deletion will result in a frame shift mutation, and it will result in change in the reading subsequent codons, and entire amino acid sequence also will be altered. You may get either a truncated protein or you may get a protein which is non-functional. So because of all these reasons, deletion is the most drastic of chromosomal alteration. Then holiday junction is observed during. So holiday junction, it is a cross-shaped structure that forms during the process of genetic recombination when 2 double-stranded DNA molecules become separated into 4 strands in order to exchange segments of genetic information. So answer is option C, that is recombination. Then coming to the next question. Huntington disease is an example of genetic disorder caused by. So it is caused by a late-acting lethal dominant allele. Now what is a dominant lethal allele inheritance? So the dominant lethal allele inheritance, the lethal allele is present both in the homozygous and the heterozygous condition. It is dominant. So naturally it will be expressed. And usually if it is expressed, what happens is the person who has got this dominant allele will not survive. Now in certain cases, only the dominant allele will be transmitted. Because at which conditions? If the dominant phenotype is expressed after the reproductive age, it will be transmitted to the next generation. Otherwise, the person will not survive, and so it will not be transmitted to the next generation. An example of this, that is the late-acting lethal dominant allele, because its onset is after the reproductive age. That is why it is called late-acting lethal dominant allele. An example of this is Huntington's disease. And it is a condition in which the nervous system gradually wastes away. People who are heterozygous for the dominant Huntington's allele will inevitably develop the fatal disease. However, the onset of the disease may not occur until age 40, at which point the affected person may have already passed the allele to 50% of their offspring. So Huntington's disease, it is an example of genetic disorder caused by a late-acting lethal dominant allele. Then next one. In a Cephedex gel filtration column, a mixture of albumin, lysozyme and thymidine was loaded. In what sequence these will be eluted from the column? So Cephedex gel filtration chromatography separates proteins solely on the basis of molecular size. And separation is achieved using a porous matrix. The small molecules have greater access to the inside of the matrix, whereas the larger molecules, they are excluded from the inside of the matrix and they will be eluted first. And the smaller molecules will be eluted later. So albumin here in the given question, it has got larger molecular size, and thymidine, it has got the least molecular size. So albumin will be eluted first, followed by lysozyme, and then the last will be thymidine. So answer will be option A. Then next one. The consensus sequence of 5' and 3' splice junctions in eukaryotic mRNA contains. So it contains GUAG. Answer is option B. This is a direct question. The next one. If total concentration of A equal T is 56%, so A and T both are occurring at 56%. What will be the percentage concentration of cytosine in the genome? So A plus T is equal to 56%. Now what will be the percentage of A? 56 divided by 2. So 28% will be A, 28% will be T. Now how much will be G plus C? Now you have given A plus T is 56%. So G plus C will be 100 minus 56, that is 44. So G will be 44 divided by 2, that is 22, and cytosine will be 22. So the answer here is option D, that is 22. Then next one. The difference between the molecular weight of sucrose and that of the sum of the molecular weights of its constituents, that is glucose and fructose, is. So first we will see the molecular weight of sucrose, that is 342. And molecular weight of glucose, that is one of the constituent of sucrose, is 180, and the molecular weight of fructose, the other constituent, is also 180. Now sum of the molecular weights of glucose and fructose. So 180 plus 180, you will get somewhere around 360. Now what they have asked, difference between molecular weight of sucrose and the sum of the molecular weight of its constituents. So what is the molecular weight of the constituents? 360. And what is the molecular weight of sucrose? 342. You must subtract both of these and you will get 18. Answer is option D. Then next one. Hybridomas are employed for. So hybridoma is a culture of hybrid cells that result from the fusion of B cells and myeloma cells. And hybridomas, they possess two properties, that is one is the property of the B cell to produce antibodies, and the other is the immortalization of myeloma cells. So, and these hybridoma cells, they are usually used in the synthesis of monoclonal antibodies. Answer is option C. Then which of the following plant hormone hastens apple ripening? So the one which is involved in apple ripening is ethylene. Answer is option D. Then next one. In bioinformatics, BLAST stands for. So it is Basic Local Alignment Search Tool. Answer is option A. Then which one of the following pairs is not correctly matched? So vitamin B12 pernicious anemia, that is correct. B6 loss of appetite, that is also correct. B1 beriberi and B2 pelarga. So vitamin B2 deficiency includes skin disorders, hyperemia, that is excess blood, edema of the mouth and throat, angular stomatitis, scoliosis, that is swollen and cracked lips, hair loss, reproductive problems, sore throat, etc. So that is the case with vitamin B2 deficiency, and pellagra is a condition that occurs due to lack of vitamin B3, also known as niacin. So vitamin B2 deficiency, I have told all the conditions like hyperemia, edema, scoliosis, stomatitis, hair loss, reproductive problems, sore throat, etc. Vitamin B2 deficiency does not cause pellagra, but vitamin B3 deficiency will cause pellagra. So the answer is option D, the vitamin B2 pellagra. Then next one, mitochondria and chloroplast carry out oxidative phosphorylation and photophosphorylation respectively by means of. So it is by means of chemiosmotic coupling. Answer is B. Then next one, the first antibody synthesized by the fetus is. So it is IgM, that is synthesized by the fetus. Then next one, the carbon dioxide concentration in blood increases, breathing becomes faster and deeper. Answer is option. Then the Ramachandran plot is relevant in the study of. So it is relevant in the study of protein structures. Answer is option C. Then which of the following compounds is an end product of beta oxidation? So it will give rise to acetyl-CoA. Answer is option B. Then one of the following is an example for an interspecific hybrid. So the mule, it is a cross between a female horse and a male donkey. So the answer, it is an interspecific hybrid. So the answer is option C, that is mule. Then next one, conversion of a floral organ to leaf-like structure is known as, it is known as phyllodes. Answer is A. An agent that causes phyllodes is, answer is option D, that is phytoplasma. Next one, the scientist who won Nobel Prize for the discovery of an enzyme is, so it is E. Buchner. Answer is option B. Next one, opines are small molecules that serve as food for. So they serve as food for, I already discussed about opines in just previous question. So the answer here is Agrobacterium species. Then next one, which one of the following is a natural auxin? Indole 3 acetic acid. Answer is option C. Then insulin is secreted by pancreas. Answer is option C. Next one, mycelium refers to the vegetative body of a or an. It is fungus. Answer is option A. Then next one, Pasture Effect refers to slowing down of the rate of glucose metabolism in Saccharomyces cerevisiae cells in the presence of. So the Pasture Effect is an inhibiting effect of oxygen on the fermentation process. It is sudden change from anaerobic to aerobic process. Answer is option D, that is oxygen. Then next one, the evolutionary process by which an organism becomes better suited to its habitat is known as adaptation. Answer is option C. Then next one, the element essential for the functioning of thyroxine is, so it is iodine. Answer is option B. Then gram stain is used to distinguish between different types of. So gram stain is used to distinguish between different types of bacteria. You have gram positive bacteria and gram negative bacteria. Then which one is an epimer of D-glucose? So two sugars differing in the configuration at a single asymmetric carbon atom are known as epimers. And if you see the structure of glucose and galactose, you see that they differ from each other with respect to OH group at carbon 4. So an epimer of D-glucose is D-galactose. Answer is option A. Next one, the instrument used to make thin section of processed tissue sample for microscopic study is, so it is a microtome. Answer is option A. Then prion. Prion is it is a protein. Answer is option D. Then dengue is caused by. Dengue is a viral infection transmitted to humans through the bite of infected mosquito, usually Aedes aegypti or Aedes albopictus. So it is a viral disease. Answer is option C. Then next one. Watson and Crick are associated with. So they are associated with the DNA double helix. Answer is option C. Pyridoxine is otherwise known as. It is called as vitamin B6. Answer is option B. Then photorespiration is the process in which Rubisco fixes. So photorespiration, it is a metabolic pathway that occurs in photosynthetic organism and release carbon dioxide, consumes oxygen and produces no chemical energy or food. So it consumes oxygen. So answer is option B. Next one. Ornithine it is a non-protein amino acid. Answer is option A. Enzyme that transcribes RNA into DNA is known as. It is called as the reverse transcriptase. Answer is D. Then which one of the following crops has its center of origin in India? It is brinjal. Answer is option C. Then next one. The high energy molecule produced by substrate level phosphorylation in TCA cycle. So during Krebs cycle or the citric acid cycle or the TCA cycle, succinyl-CoA is acted upon by an enzyme succinyl-CoA synthetase to form succinate. That is a C4 carbon compound. The reaction releases sufficient energy to form ATP in the case of plants or GTP in the case of animals by substrate level phosphorylation. Now if it is GTP that is produced, then GTP can form ATP through coupled reaction. So the answer here is GTP. Answer is option C. Then the radiation used to find bone density is. Answer is option C, that is X-ray. The next one. Suicidal bag of a cell, that is lysosome. Answer is option D. Lysosomes are called suicidal bags of cells because they contain hydrolytic enzyme. If the cell is damaged, the lysosomes undergo autolysis and burst open. This results in the release of these hydrolytic enzymes. The released enzymes then digest their own cell and ultimately the cell dies. Hence they are called suicidal bags of the cell. So that's it. Thank you for watching my channel. Please do like, share and subscribe. Thank you.